Hi everyone. So here is the second lecture in the series about plotting and visualizing data in exploratory ways. Last time we talked all about how to make plots. We also talked about how to make slightly more complicated plots where you want to plot a couple of things on top of each other, how to label the plots, how to make a grid of plots, and how to title your plots and do all of the nice things. And I motivated all of this by saying that usually what we do when we have a new data set and we don't know exactly what's in it is we begin by exploring the data by visualizing it. So today we're actually going to get a real data set and go through this process and think a little more deeply about what's going on in this particular data set and using our visualization tools and plotting tools to help us do that in this process. Okay? So one data set that I'm going to load up today is a data set that was collected actually a pretty long time ago that is available on the internet via the UCI, University of California Irvine Machine Learning Repository. This is a kind of a fantastic place on in the internet where they just have lists and lists of freely and publicly available data sets for us to play with. And I picked this particular one because it illustrates some of the nice features of being able to plot data, but very quickly things get kind of complicated. And so I think that's nice as well to realize that this is what happens with real data. Okay? So abalones. What are abalone? Now, abalone are edible snails. So if you type abalone into Google Images, you get pretty pictures like these. They are quite beautiful on the inside. People have used them for making mother of pearl. They're pretty sizable, which is why people use them for eating purposes. And so this is actually a pretty large industry in different parts of the world where they are able to farm lots and lots of abalone. And so that is the origin of this particular data set. So the data set was collected by a couple of folks in Tasmania, where they were harvesting abalone. Now, by law, as with all kinds of commercial fisheries and such, there are some regulations about ages of abalone one is allowed to and not allowed to harvest. And so they had this interesting question, which was, how do we tell how old these abalone are, right? Now, the correct way to tell it is kind of a laborious process. It's kind of like counting tree rings. It turns out that if you cut apart the abalone and then stain it, you can look under the microscope and very carefully count the number of rings it has, and that is a pretty good proxy for the age of the abalone. Now, this process has a couple of drawbacks. One is that you must cut apart the abalone in order to count the number of rings in the abalone, thus killing it. So it's not a very good way of telling whether or not you have a juvenile or an older abalone, okay, in the fishery process. The other reason they don't want to do it is because it's kind of expensive, right? You have to cut it apart, you have to stain it, you have to get somebody to send it to the microscope and stare at it for a long time. And so for a variety of reasons, they were looking for a way to estimate the age of the abalone without actually going through the process of counting the number of rings. And so what they hypothesized is perhaps the age of the abalone may be correlated in some way that may be discoverable by data science with a couple of other measurements that may be easier to make. So physical measurements that are made on the outside, for example, they're going to measure the length of the abalone and the weight of the abalone, et cetera, et cetera, et cetera, right? And so here is a data set. It's actually pretty gigantic for getting started. What they're going to do is come up with a data set, and here they says the number of instances is 4,177. So they did measurements, 7 measurements on 4,177 different individuals of abalone. Measured all of these physical attributes, as well as did a laborious ring counting exercise to figure out a proxy for exactly how old the abalone actually is. And in addition, they've also identified the sex of the abalone, whether it's male, female, or in some cases when the specimen was too young to be determined, they've labeled it an infant. Okay, so that's our data set that we're going to be working with. And so I'm going to then go back to MATLAB and load up the data set, and we'll go through the process of exploring it and seeing what we got. Okay, so we're going to go back to our working window and load up the data set. Now, the topic of exactly how to load data sets from text files is the subject of a different lecture. For the purposes of our lecture today, we're going to assume that you've already gone through the process of loading in and curating the data set. So what I'm going to do is load in a data file that I've done that process with previously so that we can just start the exploration process. Okay? So I've saved the data that I've loaded up before in a .mat file called abalone.mat. So we're going to load that up. Okay? So if I run this, it's going to load all of the variables that I've previously put into abalone.mat. So at this point is a good time to look at exactly what those variables are that I've just loaded into my active working memory. Okay? So one way to do that is type in the whos function, which tells you all of the currently active variables that I have access to in my MATLAB working directory right now. So what I have are 3 variables, x, age, and gender. X contains the actual physical measurements, of which there are 7 of them. So each row of x corresponds to one individual, and each of the 7 columns corresponds to a different physical measurement that was made. In addition, I have a number for each individual that corresponds to its age, so this is an integer, and I have its gender. So this is going to be the same size as age, so it has the same number of rows and the number of individuals, but instead of a number, I have a character. It's either M, F, or I, corresponding to the identified sex of this particular specimen of abalone. Okay, so I always like doing this first when I load up a data set. I look at what we got, what I actually have, and I look at the sizes of the matrices. Okay, so this is one of those things that's just really, really good best practice when you're working with data. You check the sizes of your matrices and make sure they make sense. Because oftentimes when something goes wrong and you have no idea what the error is, very often it's because the sizes of the matrices don't match up. Right? So if somehow your age vector is of a different size than your x vector, which contains all of the physical measurements, then you've got a problem, right? And it's a problem on the data side rather than the data analytics side. And so we always double check the sizes of the matrices first. Okay? So what are these 7 physical measurements? Well, since we got the data off of the UCI machine learning website, we're going to go back there and look at what these 7 measurements actually are. So that will tell us a little bit more about the biology of what we are actually working with. So here is the data set, and it comes with documentation about what is actually inside this data set. Okay? So we have the sex, and there are the 7 physical measurements. So what I'm going to do to keep track of it is I'm just going to write it down so we can keep track of what they are without having this window open. So I have the sex, which is a character, which is an M, F, or I, depending on if it's male, female, or infant. And then the 7 measurements are length. The second one is diameter. The third one is height. So this is a 3-dimensional abalone, so these are the 3 relevant dimensions. And then we have the whole weight, which is the weight of the whole thing. We have the shucked weight, which is the weight of the shell minus the edible part. And we have the viscera weight, which is the edible stuff that we've shucked out of it. And lastly, we have the shell weight, which is the weight of the shell after it had been dried. Okay? And so the units of these things are important, right? So we're going to keep track of the units. All of the first 3 are measured in units of millimeters. And all of these are weights, and they are measured in units of grams. Okay? So we're going to keep track of that to see what we're looking at. All right? So let's go back to our data set. And each of these 7 measurements corresponds to 1 column of the X matrix. So we're going to label them. This is column 1, right? Column 1, column 2, column 3, column 4, 5, 6, and 7 in that order in the X matrix. Okay? So we just got this into our computer, and we've never looked at this data set before. And so let's just start looking at it and see what happens, right? So let's say that, remember first of all that part of our goal was to try to figure out if we can find some kind of measurement that is easy to measure that might be a proxy for the age of the abalone. Okay, so let's just start in the beginning. Let's start with the first one. Okay, let's see how length of the abalone varies with age. Okay, so the length of the abalone is in the first column of the data set. So what I'm going to do is make myself a length variable, and I'm going to assign to it all of the rows. Remember, each row is an individual, and the first column. Okay? And what we're going to do is make a figure, and we're just going to make a plot, just like we did before with either the made-up data or the sinusoidal data, right? So we're going to plot the length by the age. And let's just stop there and see what happens, okay? So I've made use of a pretty useful function in MATLAB, which is the cell structure, okay? And that's denoted by these double percentage signs right here. You'll notice that this section has become outlined in gray, which means that it is the current cell. If I made another one down below, let's say with some other code down here, right, you'll see that as I occupy different cells, they become highlighted in turn. So the strength of doing your coding in this way is that you are able to run individual sections of your code really easily without running the entire file. So in this case, I've loaded up my file on top. I have all my data. I can run just this section without the stuff on top, just the stuff that's in the highlighted section by itself. And in the case of a Windows machine, you press Control-Enter to run just this section. And in a Mac, it is a Command-Enter, and does exactly the same thing. Okay, so we're going to run just this section in here, which is define the length and then do a plot of the length versus age. Let's see what happens. Okay, that is a giant mess. Okay, think about it for a second. What happened here? So just like with the plot that we did last time in the plotting section of the lecture, the default behavior of the plot function is that it takes every point, it plots the point, and then it draws a line from this point to the next point. Now, because every point here is an individual and these are all independent samples, they come in some random order. And so if you plot lines between individual 1 and individual 2, and plot another line between individual 2 and individual 3, you'll end up with a kind of a jumbled bird's nest looking thing, like over here. Okay, so it's not what we want, but it's exactly what the plot function does by default. So if we don't want this, we're going to have to go back and modify it so that we plot something a little more sensible. Okay, so let's get rid of that. And remember, we can have a couple of optional parameters of the plot function in order to make it not do the things that it does by default. And one of the things we can do is plot using a different set of markers, and don't use lines to connect them, because that's what we want in this particular case. So let's say we want x's. We're going to have an x for every data point. Okay, so we have an x, and we run that. And that looks a lot better, right? This looks a lot more like what we actually want to see. So we have length on the x-axis and age on the y-axis. And so let's make sure the units make sense, right? The length is supposed to be in millimeters, right? And so the middle of this range is like 100 millimeters, which is 10 centimeters, which is about yay big. So that makes sense. That's how much a big abalone, a big edible sea snail would be. And the age is in years, right? So it goes from 0 years to the oldest sample they had was almost 30 years old, which is pretty impressive for a sea snail that's been harvested. Okay, so let's go ahead and label our plot in that way. So the X labeled properly would be length in millimeters, and the Y label would be age in years. Okay? There it is. Great. So one of the great features of exploring data in a programmatic way, rather than kind of doing everything by hand as you think of them, is that you can do a lot of things really quickly. So for example, we know that the age looks like it's positively correlated with length. That's good. But what about all of these other parameters? How does it vary with all of these other parameters, right? So what we can do is very quickly make a bunch of these scatter plots and look at how age varies with every single one of these parameters in a quick way, okay? So for this, we're going to make a couple of subplots and just try to plot all of this data in a big pile and just look at it, because that's usually a good place to start, okay? So here we go. I'm going to now go to the second cell. I'm going to leave the first cell and just going to do something slightly different, okay? So for here, I know I have 7 measurements. So I'm going to make a variable called n, which is going to be the size of x in the second direction, okay? So remember, the size of x returns 2 numbers, number of rows and number of columns. Okay, the number of rows in this case is the number of individuals, the number of columns is the number of measurements. So I want that second number because that tells me how many different kinds of measurements that I've taken. So perhaps I can name this a little more informatively and call it n measurements. Okay, so then what I'm going to do is I'm going to make a figure, and I'm going to make subfigures. That's going to tell me how each of these different physical measurements varies with age. So let's do a for loop for I equals 1 to n measurements. I usually like typing the end right when I type the for loop so that there's always the beginning and the end and I don't mess up. And we're going to do a subplot. So since we have 7 things, we're going to have to round up to the nearest number that's not prime. So let's do 8, which means we can do a subplot with 2 by 4. Okay? So 2 rows, 4 columns, and we're going to use the ith element of that subplot. And we're going to do a plot of the ith column of x, which corresponds to each of these physical measurements in turn. And we're going to plot that by age, and we'll want a little x at each of those dots. Okay? So let's plot that. And here is the plot that we end up with, okay? So each of these in turn, and I can label them if I wanted to because I know exactly what the measurements are. So here's length, diameter, height, weight, shucked weight, visceral weight, and shaw weight, each in turn. And on the vertical axis, we always have age, okay? So there's a couple of things that are obvious from looking at the data in this way. Okay, this is by no means the only way of looking at it. It's simply one of the easiest ways to look at it, so I do it first, right? So observation number 1, I'm just talking here by looking at this plot, is that most of these measurements are positively correlated with age. As each of these numbers, physical measurements, gets bigger, age also gets bigger. That makes sense, right? Most animals grow bigger as they age, so that's all good so far. And I'm saying this sort of as a way of double checking, right? When you get a new data set, if you collected it, you kind of have more of an idea what it is. If you did not collect the data and you get it either from a collaborator or from somebody's primary publication, then you download their data from the internet, or from more of an open public data set like this that we've done from the UCI machine learning database, you don't actually know what the data really is. And so one of the first steps is that you look at it, you use whatever intuition and knowledge you have about the data set to make sure you know what you're looking at and it actually does make sense, right? That's just a way of double checking and making sure you're not doing something that is foolish because of lack of experience with that particular data set. So this is always a good exercise to do, even if it seems a little bit silly, right? So a couple of things that we can note. One is that they are positively correlated. That's good. That makes sense, right? The other thing we note off the bat is that it's really messy, right? There is not a simple, clean, linear relationship between any of these parameters and the y-axis, which is age, the thing that we wanted to predict, perhaps, right? So using any of these alone is probably not going to produce great results because the data is pretty messy. Okay. So this motivates us to use more sophisticated methods, and that's going to be the subject of a lot of what we're going to be talking about in the remainder of the course. Both how to explore this type of data, potentially messy data, and the messy data is really, you know, perhaps it's not what you expected, but it's usually an interesting challenge, right? We use increasingly larger hammers to solve the problem of how to actually predict the age of the abalone based on these 7 measurements that we have. Okay? So that is the first thing that we do in terms of exploring this data set, this brand new data set that we've just opened. And we'll continue exploring it in the next section.